Hello friends, welcome to Simplified Biology. Today's topic, as requested by many, is DNA structure. DNA and RNA are nucleic acids. Nucleic acids were discovered by Frederick Miescher. Nucleic acids are polynucleotides, means polymer of nucleotides. And nucleotides are made up of nitrogenous base, pentose sugar and phosphoric acid. It is phosphoric acid due to which nucleotides are negatively charged. Hence, nucleic acids are negatively charged, or you can say they are polyanionic. Nitrogenous bases are of two types, pyrimidine and purine. Pyrimidine is cyclic. It is just like a benzene ring that has 6 carbon atoms. In this, of the 6 carbon atoms, 2 are replaced by nitrogen atoms. Pyrimidine are of 3 types, thiamine, cytosine and uracil. Purine is heterocyclic and is formed by the fusion of the pyrimidine ring with an imidazole ring. And purine are of 2 types, adenine and guanine. In DNA, adenine, guanine, cytosine and thiamine is present, while in RNA, adenine, guanine, cytosine and uracil is present. Thiamine is methyl uracil and it is more stable than uracil. Pentose sugar is a 5-carbon sugar. It is ribose sugar in case of RNA and deoxyribose sugar in case of DNA. The difference between ribose and deoxyribose is absence of oxygen at the 2' carbon of the sugar. It is due to this that deoxyribose sugar is more resistant to hydrolysis than the ribose sugar. Nitrogenous base plus sugar form a nucleoside. Nitrogenous bases are attached to the sugar by glycosidic bond. Glycosidic bond is formed between the first carbon of sugar and first nitrogen in case of pyrimidine and nitrogen at 9th position in case of purine. So these are the glycosidic bonds formed between the base and the sugar, forming the nucleoside. Nucleoside plus phosphoric acid forms the nucleotide. Now the phosphoric acid is attached to the 5' carbon of the sugar by an ester bond. So phosphoric acid is attached to the 5' carbon of the sugar to form a nucleotide. Now how the nucleosides and the nucleotides are named. Nitrogenous base adenine, its nucleoside is named as adenosine in case of RNA and deoxyadenosine in case of DNA. And its nucleotide is named as adenylic acid in case of the RNA and deoxyadenylic acid in case of DNA. Or you can also refer them as AMP, adenosine monophosphate. Similarly, guanine is referred as guanosine or deoxyguanosine. Its nucleotide guanylic acid or deoxyguanylic acid. Cytosine referred as cytidine or deoxycytidine. Its nucleotide cytodylic acid or deoxycytodylic acid. Thiamine, which is only present in DNA, hence deoxythymidine, and its nucleotide deoxythymidilic acid. Uracil, which is only present in RNA, is uridine and uridylic acid. Now, adjacent nucleotides are joined to each other by phosphodiester bonds. Remember, nucleosides are joined to each other by phosphodiester bond, which is formed between the 3' carbon of one sugar and the 5' carbon of the other sugar. 3' carbon of one sugar and 5' carbon of the other sugar by the help of phosphodiester bonds. In a polynucleotide, adjacent nucleosides are held together by phosphodiester bond. Now, there is always a free phosphate group at the 5' carbon at one end, and this is referred as the 5' end. And there is a free hydroxyl group in the 3' carbon at the other end, and that is referred as the 3' end. So now come to the DNA structure. DNA structure was given by Watson and Crick, for which he received the Nobel Prize. Now, his discovery was based on the discoveries given by Wilkins and Franklin, who by X-ray diffraction method gave the helical nature of DNA, Estbury, who gave the three-dimensional structure of DNA, and on Chargaff's base equivalence rule. Now, according to Chargaff's base equivalence rule, in a DNA molecule, amount of purine is equal to pyrimidine, amount of adenine equals to thymine, amount of guanine is equal to cytosine, and the A plus G by T plus C ratio is 1. The A plus T by G plus C ratio is constant for a species, and it differs in different organisms. This led to the discovery of the base pairing found between the nitrogenous bases, an important step in the structure of DNA. A DNA molecule is made up of two polynucleotide chains. Now, these are the two polynucleotide chains seen in a DNA molecule, where the sugar and the phosphate forms the backbone of the DNA, while the nitrogenous bases project inside. DNA is a double-stranded, means consists of two polynucleotide chains, right-handed, helically coiled structure, where the two strands are held together by hydrogen bonds formed between the nitrogenous bases, where A always pairs with T, double bond, while G always pairs with C, triple bond. There are two hydrogen bonds between adenine and thymine, and 3 hydrogen bonds between guanine and cytosine in a DNA molecule. Now, the two strands in the DNA molecule are antiparallel, means they run in opposite direction. If one is the 5' 3' strand, the other one is the 3' 5' strand. This is in relation to the position of sugar in the two strands. In one strand, the sugar is present this way. Its 5' carbon is towards the upper end and 3' carbon is towards the lower end. This is the 5' 3' strand. While in the other strand, the ribose sugar is present this way, where the 5' present below and the 3' present above. This is the 3' 5' strand. The two strands are complementary to each other. Now, this complementary to each other means if you know the base sequence of one strand, you can find out the base sequence of the other strand. Means if the sequence of one strand is GACC, That means the sequence of the other strand is CTGG. This is possible due to specific base pairing. Where always A pairs with T and G pairs with C. Diameter of a DNA molecule is 20 angstroms. Space occupied by a turn, this is a complete turn, is 34 angstroms. There are 10 nucleotide pairs in a turn, means space occupied by a nucleotide is 3.4 angstroms. The nitrogenous bases are at right angles to the long axis of the helix, and they are at an angle of 36 degrees with respect to the adjacent bases. The helical nature of DNA is possible only due to the rotation shown by the glycosidic bonds formed between the nitrogenous base and the sugar, while hydrogen bonds provide stability to the DNA molecule and phosphodiester bonds provide rigidity or stiffness to the DNA molecule. There are 5 types of DNA, A, B, C, D and Z, of which bDNA is the native type. Length of DNA is defined by the number of nucleotide pairs present in it. As we know, the size of each nucleotide pair. In bacteriophage, that is I into 174, there are 5,386 nucleotides. In lambda bacteriophage, there are 48,502 base pairs. In E. coli, 4.6 into 10 to the power 6 base pairs, while in a human haploid genome, there are 3.3 into 10 to the power 9 base pairs. So this is all about the structure of DNA. Thank you for watching.